All right, good afternoon, everyone. It's my great pleasure to introduce Caleb Bashor from Rice University. Once upon a time, Caleb was a Reedite, that is a Reedy, someone from Reed College. And in his undergrad, Caleb became essentially a super hardcore biochemist, doing things like working out ping pong reaction mechanisms and doing some really hardcore stuff. And after that lifestyle, he decided to pursue his PhD at UCSF with our shared interlocutor, Wendell Lim. And Caleb was a senior grad student in the lab when I joined there, and he was really a towering figure in the lab. And that's not just because of his extraordinary height, but also his extraordinary influence he had on the lab and the students like myself who joined there. And one of the things that Caleb really impressed me by when I was a student was his ability to take his knowledge and understanding of biochemical principles of molecules and use that to shape his sort of synthetic biology pursuits. And this enabled him to really start sort of rewiring signaling pathways in extremely novel ways using engineered scaffold proteins, something very cool to come out of that work. He then did his postdoc with Jim Collins, where he can kind of continue to apply his dual wielding of biochemistry and synthetic biology, this time turning his attention to a variety of interesting questions, but synthetic transcription factors and gene circuits. And this led to really, I think, substantial gains for the field in terms of developing these tools, but also insights into biochemistry, in particular, ideas about cooperativity and its roles in biology. And since starting his own lab, Caleb has been pursuing new methods to allow high throughput construction and characterization of both genetic and phosphorylation-based circuits. And so rather than hear me, let's just let Caleb take it away with what will be a great seminar.
Oh, wow, the room really filled out. Am I projecting?
Sounds like I am. Okay, great. Yeah, thank you for the intro, Scott.
It's nice to talk to a biochemistry department. I don't talk to many biochemistry departments these days. I've gotten lost in the world, the community of bioengineering over these last 5 years in my lab. There's a lot of emphasis on translation in bioengineering. And so I really try to toe the line between doing what we call foundational synthetic biology, which really just means figuring out how to make new types of circuits. And also trying to translate. And I think these last 5 years in the lab have really been about trying to teach my mostly bioengineering students basics in biochemistry. Where do interactions between proteins come from? What is it that's really useful to think about in terms of protein properties when you're building circuits? And I think going back a long way, this is something that Scott and I both noticed, because we were in a lab that was kind of on the ground floor in the early days of synthetic biology and kind of the early 2000s, and they're really getting circuits to work, building synthetic gene circuits, protein circuits, whatever. You have a real advantage over other folks in the field who are coming from other disciplines, if you think about things as a biochemist or a biophysicist would. So, I think all of you, whether you know it or not, have an intrinsic tool set. I assume most of you are biochemistry students. Maybe that's not totally true, but all of you guys have an intrinsic tool set for building synthetic systems, which you should feel free to exercise at any point in your career. Okay, so my lab, kind of me going back a long way, and now also my lab are really interested in how natural systems are organized and how cells use protein networks to sense and process and respond to signals in their environment. And so it's very much our goal to take the natural design principles that we see in cellular systems and try to convert these into engineerable, tunable regulatory interactions that we have control over, that we can connect to inputs and outputs of our choice. And we work with both signaling and gene circuits. We're sort of agnostic to what type of regulatory networks we work on. And at the end of the day, we're trying to control cell behavior. And I think this is kind of what I mean by fundamental synthetic biology is, being able to build new types of regulatory connections that give you purchase on behavior that you might want to control. But, maybe there isn't a good engineering framework for controlling it yet. And, there's still a lot to be done in synthetic biology. The window is narrowing. We're getting better and better at controlling, new types of cell behavior, but there's still a ton to do in human cells. And I think so that's kind of where I decided to start my research program is, working with human cells. And, we really think, our view really is that if we can establish robust, tunable connections between, for example, the outside of a cell and regulatory processes in the inside, then we can build really cool and powerful biotechnology. And so, part of my group is focused on foundational circuit engineering, and then part of it is also, looking toward translation. And this is a vision that we're really motivated by, and this is by no means unique. There's a lot of people in the syn biofield that have been sort of inspired by this vision for a long time, and that's one of a cell-based therapeutic controller. And so this is something that can, sense biomarkers associated with disease states, sense signals associated with human physiology, process the signal via some synthetic circuit, and then actuate on the patient, maybe through some secreted therapeutic. So the advantage to this is, this cell-based controller can, in principle, embody all of the things that, say, are distributed across different biomedical devices, the controller function, actuation, the sensor, and even communication potentially with external devices can all potentially be embodied in engineered cells. And so I think this is an exciting, and then you can take those engineered cells and actually place them proximate to human physiology. So this is something that we're going to try to build toward, and again, many others in the field are trying to build toward, over the coming years. But the challenge is, what goes in this? What goes in the circuit here? What's the best way to connect these different functions and embody this? And the thing that we always bump up against is, how to design, how to build, and how to navigate this design, build, test, learn cycle. And so I think that may be less familiar to this audience than it is to engineers, but it's something that we kind of have to engage all the time when we're building circuits. So, so design, build, test, learn in synthetic biology, it really starts with a mechanistic understanding of what you're working on, the system you're working in, and some idea of where modular genetic parts that you're going to use to build your system, to build your circuit come from. And then there's going to be some sort of quantitative model or theory that gives you an idea, a quantitative idea of how they're going to work when you put them together and put them into a cell. And so that's the ideation phase. And then you have this idea of what you want to build, how it's going to work. You embody it in DNA, you put it into cells, and then you test the cells. And an initial test of a circuit is usually you're looking at some controlled input, and then you're measuring the output. And you're looking at that transfer function, and you're trying to see whether or not it matches up with your original hypothesis. Okay, almost never does, right? The first time, nothing, it barely works. And so the real art of doing this in the first place is just to see anything work at all. Okay, and so this is what a project looks like in synthetic biology. And we can be optimistic and say these are experiments. These are really more like these individual steps within a genetic design space may be more like graduate student years. So you have an initial design, you're trying to get to a target behavior. Maybe in the worst case, these are dissertations. But this is something where as we try to pursue challenges that are on the frontier, we've always been just kind of bumbling through design space. And I think the folks in the bacterial side are better at this now. This is how a lot of the projects in my lab have gone. This is how a lot of projects in my career have gone. And believe it or not, this is a publishable result. Many of these are actually publishable results. But so why is this so tough, right? Well, so I'm going to give historical reasons here, because I think it sets up the rest of my talk really well. One is, just a lack of genetic parts. Where do we get parts to build stuff from? What sources do we draw them from? And, the question is, goes back to biology being modular. Where do we see the modules in biology that we can draw genetic parts from to build our circuits? Okay. Second one is, even though parts seem to be modular, on paper and in theory, they're not fully modular almost ever. Parts are context dependent, right? And it's because they're molecules that exist and coexist in a crowded environment. They interact with other parts in the circuit. They interact with things in the host cell. So anytime you take a design that you've made in one context and move it to another context, it's almost always going to change function, okay? So this complicates things. What this adds up to is it's really hard to develop reliable models for building, okay? And I think that there's really been two solutions to this problem that have been sort of articulated. And one is to do the design, build, test, learn cycle smarter. Okay, and I think this is the one that most people have embraced in the last decade or so. And there's an investigator named Chris Voigt at MIT, who some of you might have heard of. And he's really kind of written the book on building design frameworks that are smarter, that are more modular, such that when you construct a system, it's easier to model and predict their behavior, okay? But there's a downside to that. You lose some functionality. So these systems end up maybe not looking exactly like natural systems look, and maybe they don't have the same functionality. So advantages and disadvantages. And then there's the other option is just to do more. And there's companies that are able to build many hundreds or thousands of constructs and test those in parallel in a way that we can't possibly do in an academic lab. And I think that if we were able to all have the resources that are at their disposal, I think we'd all like to work on our projects at the same throughput as they do, but it's not feasible. So I think there's a lot of emphasis now, kind of more and more on figuring out ways to do things bigger and faster, but also for cheaper. Okay, so the talk is basically going to be about two different projects that both kind of embody these two solutions. All right, and they are the following. So first one, I'm going to talk about engineering synthetic phosphorylation signaling circuits and cells. So here we're really developing a framework that falls under that first solution. And again, I'll show you the pluses and the progress we made, and then also the minuses and the limitations with this approach. And the second one is kind of a more general approach, and this is something that we did recently, kind of just sort of spun up in the last 3 years in my lab, where we've developed a platform where we can build many circuits, hundreds of thousands to millions of circuits in a single experiment, test them all at once. And now we started to think about how to use machine learning driven circuit engineering as sort of the way to, not necessarily supplant biophysical models and biophysical thinking, but maybe to complement it, okay? So I'm gonna start with talking about this, and I'm gonna motivate it with some work I did in my postdoc. And I'm gonna be talking about the SynTF platform here, which is a way of engineering synthetic circuits or synthetic gene circuits in eukaryotes using C2H2 zinc fingers, and really relying on the programmability of their binding to target sequences to very easily engineer transcriptional connections. Okay, so the zinc fingers as engineered transcription factors are fantastic. If you guys ever want to build synthetic gene circuits in 1 million cells or whatever, you guys should absolutely look into this platform. The zinc fingers are very easy to program. You can add domains to either the N or the C terminus. You can get these things to interact with other proteins within the transcriptional complex. You can get them to activate transcription by interacting with core promoter proteins. And as a result, it's really easy to program transcriptional network connections with these things. Okay, so we published a bunch of papers on this going back a decade now. This is work that I've led and has been in collaboration with mostly with Mo Khalil at Boston University, who's really the leader in this space. And I just want to give you guys one demonstration of kind of the programmability here. And this is a paper that kind of came out around the time I was starting up my lab. And it's in yeast. I've moved away from yeast, but I still think yeast is kind of the best synbioorganism to work in. Just because, again, like the design, build, test, learn cycles are just, they're quicker. And then you have a lot of freedom to, so yeast transgenesis is the best. You can put things in a single copy. You can build libraries in yeast, absolutely fantastic. I really wanted to start my lab by making mammalian cells like yeast, and we've made some progress there, but it's still kind of an ongoing challenge. But so what we did was we put some simple 2 and 3 node circuits that have these SynTFs in them into yeast. We programmed the zinc finger binding to the different transcriptional or the different promoter complexes. And the goal was here to ask if we could build circuits that are actually able to decode information in the frequency domain. So these are in microfluidic devices. And then we're sending different amounts of inducer or sending different frequency of inducer inputs at them. And so this is all basically model-driven engineering of synthetic circuits. We need a low-pass filter and a band-stop filter that were able to activate in specific temporal environments. So I was very shocked that this worked. I was very shocked that this worked as well as it did. And why did it work so well? Okay, so well, it comes down to the parts that we were building with. And the thing that's nice about the SynTF system is simple biophysically defined parts with interactions that we could actually measure in vitro. And then we could put these into cells. We could build models based on those interactions where the behavior of the circuits was largely predicted by the models. And the models were simple too. It was just thermodynamic interactions between the proteins and then with the DNA binding motifs in the promoter. And as a result, we could do something that is kind of for many synthetic biology projects, kind of a holy grail, where we could take all of the different combinatorial part diversity that we can imagine, and we could kind of predict what the behavior space looked like, and then we could kind of select things from that behavior space to build that were target behaviors, and mostly get it right. So, I mean, that was great, and that's kind of to this day sort of the only system that I've really worked on that kind of meets those standards. But there are certain disadvantages to just working with the transcriptional system. And so, going back to this idea of the cell-based controller, what kind of circuits would we need to realize this vision? Well, physiological timescales, things that happen on minutes timescales to hours timescales, you need something faster than a gene circuit. Gene circuits turn on in a matter of hours. In human cells, it sometimes takes a day or multiple days for circuits to turn off. So the time resolution for gene circuits is good for some applications, but if you're trying to monitor physiology, maybe not so good. So what we really need is something that senses and responds to events on a seconds to minutes timescale. Okay, so that's going to be post-translational circuits. Transcriptional circuits are not adequate. We need something that acts on a faster post-translational timescale. So that's going to be this first project I'm going to talk to you about, where we built synthetic signaling circuits. Really trying to take all of the best parts of the SynTF system, or the best aspects of the SynTF system, and why they worked, and incorporate those into phosphorylation-based signaling circuits. So we developed a design logic, a composable molecular parts grammar that allowed us to do this, I'm going to tell you about. And as I'm going to show you, we could create customizable connections between upstream inputs, extracellular inputs, and then downstream intracellular outputs that operate on a fast timescale. And this is very much a work in progress. There's stuff that I think we're going to be able to continue to do with this system for many years to come, but I'm going to show you kind of the fundamentals of why I think it's working well, and why I think it's scalable. And I want to give a shout out to Xiaoyu Yang, who is a graduate student that joined my group in 2019, who is absolutely fearless, probably one of the best experimentalists I've ever seen work. He can do up to 3 or 4 extremely high quality experiments in mammalian cells a week, and still go home and cook dinner. Extremely talented individual. This paper is under review right now. It's on bioRxiv, so please take a look. And so the system we developed is basically based on 2 design strategies that sort of go together. And the first is the recognition that pretty much all phosphorylation signaling pathways are built up from individual enzymatic amplifiers. So we call these push-pull motifs. This is This is a term that Sharad Ramanathan came up with many decades ago, and or 2 decades ago actually, and I think it's cute. So we call these push pulls. So push pull is a kinase, a phosphatase that makes a phosphate mark and then erases the phosphate mark, and then the substrate protein that gets interconverted back and forth. So we thought if we can make push pulls and we can connect push pulls together, then we could capture a lot of the systems properties that we see in signaling pathways. And so we're going to build these push pulls using basically individual protein domains. So catalytic domains, kinases, and phosphatases taken from phosphotyrosine signaling in mammalian cells, and then interaction domains. So for obligate interactions, where things just kind of always come together, we're going to use leucine zippers. And then for conditional interactions that are phosphorylation dependent, we're going to use SH2 domains. Okay. And then, of course, phosphorylated motifs that are phosphorylated motifs that are recognized by the kinase and the phosphatase, and then also bind to the SH2. And as I'm going to show you, the phosphorylation or the phosphorylation and binding by the SH2 is the thing that we're going to be able to use to connect push pulls together. All right. And so I just want to go through and describe how we build and measure these. And so we start off by just taking kinase and, for example, here kinase and substrate on plasmids, transfect them into hex cells. And so these are going to express as kinase and substrate domain, and the interaction between the kinase and the substrate is going to be mediated by a leucine zipper. Okay. And so by virtue of this recruitment, this is going to give you the phosphorylation on the substrate. Okay, so we can read these out with fluorescent antibodies. Okay, so there's epitope tags on the kinase and the substrate. We also have an antibody against the phosphorylation event. So we can kind of take a look at everything that's going on with the system on a single cell basis, which ended up being really powerful and really enabling for both building stuff and then also modeling it. So here's a plot, and these hexagons are basically different size hexagons at different density of cells here. Okay, so kinase and substrate expression. And so you see we transfected the cells. So cells up here have more kinase and substrate. Okay, and then we're going to overlay on that the color map, which is phosphorylation. Okay, so you see this makes sense. More substrate, more kinase in the cells, and you get more phosphorylation. So we call this a hexagonal hit and heat map. This is a triple H map, just like the wrestler. And this is going to be kind of how we describe all of the how I describe all of the data for kind of the rest of the presentation. Okay, and so when we put all the triple H maps up here for the recruited kinase and then the controls, what we see is the recruitment of the kinase is the thing that makes the phosphorylation happen. Unrecruited control, non-phosphorylatable control, no kinase control, and then also kinase dead control. We don't see any phosphorylation. Okay, so the phosphorylation here is recruitment dependent, which kind of gives us the interactions that we need to start to wire together the pathways. Okay, so we can also add in phosphatase here, and oops, sorry about that. We can add in phosphatase here, and we can see the phosphorylation go away as a result of expression of the recruited phosphatase. Okay, so here we have an intact push pull. So because we have the important biophysical parameters, the rate of the phosphorylation, and then also the interaction between the kinase and the substrate embodied as individual domains, we can tune these properties individually. So we can have different affinity zippers, different activity alleles for each of these domains. And then we can also express these at different levels. So this gives us a tunability of this gives us a tunability for the push-pull that allows us to first fit a limited data set of push-pulls and then kind of project the rest of the part space using a modeling scheme. And this is kind of the thing I told you about before that we could do with the SynTFs. Okay, so these red guys here are training set for fitting the model. And then everything gray here are individual push-pull part configurations that have different levels of phosphorylation, okay? And so we look at the phosphorylation level from the model, and then we compare that to actual data, and there's relatively good agreement. Okay, so this has, at least some of the features that our SynTF framework had, where we're able to, manipulate individual biophysical parameters and then get different predictable. In this case, it's not quite a circuit; it's just a single push-pull, but we can tune the behavior of the system. Okay. So then we can now connect these push-pulls together into networks, and we do that kind of as follows. We make the substrate from the push-pull, from the first push-pull, now the kinase in the second push-pull. So first phosphorylation happens. You get a phosphorylation here that is now recruits an SH2 containing substrate that's in the second push-pull, and now this phosphorylation happens. Okay, so 4-protein system, put this into hex cells, and again, so here we see in the presence or absence of, in the presence of the upstream kinase, you get phosphorylation. Absence, you get no phosphorylation or very little phosphorylation on substrates 1 and 2. And then we can do that trick again now with our model, where we project all of the different part combinations and what the behaviors are. And here we're really interested in looking at what would refer to here as the high gain configuration behavior. So things that amplify signal from the top of the cascade to the bottom of the cascade. So this is plotting phosphorylation with this upstream kinase versus without it. And so this is the region where you kind of get the most stoichiometric amplification from the top of the cascade to the bottom of the cascade. And then we go through now and we look at, using the model, look at some of the things in this high gain space. And the model actually does, again, a reasonably good job of recapitulating these. So we can now pick out 2-step modules that will give us good amplification between the first step and the second step. And that'll come up later when we're trying to build this into pathways. Okay. Okay, so this is by and large a validation of the design strategy, where we've basically demonstrated that we can take push-pulls. Push-pulls can be expressed, tuned, and interconnected in cells. And so now we want to try to actually do useful things with them. And so the first thing we did here was we connected them to upstream receptors. Okay, and so here the way this works is there's two receptor halves in the membrane. And they're going to come together when they bind to this chemical dimerizer. And they're going to form this complex where substrate can be recruited to one half of the receptor and then be turned over by the kinase. And then, of course, be turned back by the phosphatase there. Okay. And so this works really well. When we add in the dimerizer, we see really high fold change, 20-fold change. I wasn't expecting that with a system like this when we have the phosphatase in there. And so then we asked, like, okay, so that was a steady state measurement taken at 24 hours. So then we asked, okay, now how fast are these pathways actually going? And to test that, we devised the following system, where we express this IDP tagged M-cherry, which inside of cells forms these condensates. Okay, and so what we're looking at here is when the phosphorylation happens, the accumulation of the phosphorylated substrate in the condensate with the M-cherry tagged SH2. Okay, so you're looking for, upon phosphorylation, a change from a red condensate to a yellow condensate. Okay, and so let's watch that happen. Whoa, that goes very fast. Yeah, so you see, like, this turns on relatively quickly. You can start to see activation of the pathway within 5 to 10 minutes. It's going to reach a steady state after about an hour or so. Probably because most of that's how long it takes for the substrate to be completely phosphorylated and then go into the condensate. And then you see when we add an inhibitor for the kinase here, the off rate for the system is relatively quick too. So you compare this to other native pathways, it's actually pretty comparable. And I was very surprised by this. I thought these things would be ridiculously slow to start with, but kind of in the first version here, we're able to get pretty close to cytokine signaling pathways. not really quite able to match the speed of EGFR signaling, but that happens ultra quick. So maybe someday, maybe we're on the way to that. But relatively fast response for synthetic system, and certainly much faster than the transcriptional system. Okay, so we went ahead and made different types of receptors, connected different types of inputs to our signaling pathways. So now, we have a number of these now. I'm showing you kind of the best one at the moment, which is the TNF alpha receptor that we engineered. We've engineered a number of GPCRs to hook up to our pathways too. And this is just a quick, I mean, I'd love to get into this a little bit more, but just kind of quickly going over this. This is one of the really kind of unique applications we found for hooking these pathways up to different types of physiological responses. And this is something that John did where he hooked up the substrate to something that, so it's actually a protein binder for a calcium channel that when it binds, it actually opens the calcium channel and then allows vesicles that are loaded with a cargo that you can pretty much define as you like to be secreted from the cell. So this secretion, I don't have the plot here, but this secretion happens pretty quickly, like within a couple minutes. So this system has to be refined, but what we have here potentially is the ability to do really fast timescale secretion using the synthetic circuit. And so the system that kind of ended up as the final figure of Zhao Yu's paper was the following, where he built an entire signaling pathway that went all the way from the membrane to the nucleus. So this is 7 genes, 6 proteins, where you have the receptor turning on this intermediate kinase, and then this intermediate kinase is actually shuttling back and forth into the nucleus, and that turns on it actually turns on a SYNTF in the nucleus, attracts an activation domain, and turns on transgene expression. So reasonably good fold change here from optimized version of that pathway. And then we took the pathway and then we hooked up a TNF sensing input, and then we made it express IL-10. And we asked if we could start to move toward that cell-based controller vision that I showed you guys in a couple slides earlier. And so in this demonstration, we're programming the cells to sense TNF alpha that's being secreted by activated T cells, and then secrete IL-10 in response, which is going to turn down T cell activation and stop their proliferation. So you can think of this as kind of like a cell-based anti-inflammatory drug, potentially. And so this is done in trans-well co-culture. We add the activated T cells and the trans cells containing the transfected circuits. They exchange their cytokines, and then we sample over time. And what we see is when there's no circuit present in the hex cells, we see kind of runaway train TNF alpha being secreted, lots of T cell proliferation. When the cells just secrete IL-10 constitutively, we see this accumulation of IL-10, which is can also be something that is toxic to a patient being administered an anti-inflammatory treatment. And then when we have the circuit intact, what we see is both of the cytokines kind of hit a low set point and then just kind of persist at that low set point. So circuit works, and certainly we could improve the performance by reaching the set point earlier or having much lower having a lower set point. But for all intents and purposes, this was again very surprising that this worked kind of as advertised, but we're very happy with the result here. So just to summarize this part of the talk, we established a platform for post-translational circuit engineering in human cells using phosphorylation. And I think this is kind of not so much a first-in-class demonstration for the field. There's been other synthetic phosphorylation papers, but I think this kind of gives people a framework to start to work with and think about how to design stuff further. And I do hope it catches on, but if nothing else, we've shown that it's feasible to take kinase domain parts, use this framework of building push-pulls and fitting them together, and kind of think of this push-pull motif as now the kind of the fundamental unit for engineering signaling circuits in the same way that a trans-regulated promoter is for gene circuits. We're hoping that this catches on. We're capable of making diverse network connections to inputs and outputs. And there, because this, the currency of connection is really just about bringing things together via recruitment. There's this possibility that we can kind of connect anything outside of the cell to anything inside of the cell. And exploring a lot of options with collaborators on how to kind of put that into practice to build circuits that they want to see. And then, of course, there's a lot of potential for biotech here. And, part of the challenge there is getting these into primary cells or getting these into cells that can go into a therapeutic pipeline. And that's also an ongoing area of work in the lab. So the next steps are, these are kind of current projects in the lab. We're diversifying receptors. We're building receptor libraries to try to explore the use of other inputs for driving these circuits. And we're doing kind of some of the same things that folks did in the early days of gene circuits and exploring different topologies and just kind of asking basic information processing questions. When we have different numbers of nodes and connectivities between the nodes, what types of signal processing behaviors can we get? And something I'm really excited about, which is still a little bit out there, is now engineering these things to turn on different in vivo imaging modalities. And there's a couple examples here, just kind of really early things that we've gotten to work or are exploring. For example, here this ferritin condensate. So ferritin is something that, as some of you may know, sequesters iron. And it turns out when you condense ferritin, it produces an EPR signal. So this sense and condense response can actually generate a change in EPR signal that we can detect potentially with a wearable device. And I have a collaborator working on trying to bring the wearable technology together with our cell-based approach. Okay. And then, of course, we're also trying to get these to work in primary cells. And one of the early successes is getting to express the circuits in mesenchymal stem cells, which is one of the types of cell-based therapy platforms that we work with in our lab. Okay. So just change gears now and talk about this ultra high throughput mapping of design space, genetic design space platform that we've been developing over the last couple of years, and just kind of getting back to the challenges with the DBTL cycle. So, I mentioned that, solution one is really about trying to bridge the gap between these. Solution one is really about trying to bridge the gap between these three difficulties, right? Trying to make things in a smarter way using a framework that can be more engineerable. And the other thing that we wanted to explore, which is, by no means mutually exclusive with solution one, is just to make a lot in parallel at once. And I think part of the inspiration here has been, the kind of trying to aim at the goal of unlocking AI and ML. And we've seen this do so much recently in the protein design space that, the potential for having this be a solution or, a route to solving some of the problems we see with genetic circuit design is really exciting for me. But the challenge is the data sets are simply not available. So I think one of the other aspects to this work I'm going to tell you about is it gives us a means to generate the data sets that we could potentially use to train ML models. And actually next slide here. But so this is already something that's seeping into synthetic biology in a way that is kind of impacting just mostly the design of genetic parts. And so you think about these things in terms of sequence to function challenges, where you have a genetic part sequence and you're diversifying it using a massively multiplex reporter assay of some sort. And then you're looking for rules for how you're training an ML model to tell you what the rules are for designing part sequences. So example from Jim's lab is programming RNA switches. It came out a couple of years ago using a deep learning model. And this more recent one from Georg Selig's lab. And I don't think this paper is out yet, actually, but it went up on bioarchives about 6 months ago or so. But this is trying to understand sequence determinants for the 5' region of UTRs in therapeutic RNA. So all of these are great for individual genetic parts and things that kind of fall under the, the 500 base pair or so length limitation of Illumina sequencing. But most of our gene circuits are longer than that, right? So that's, usually, some of the cases in the earlier part of the talk I showed you, circuits that were like a full 20 kb in length, right? So how do we deal with those? How do we get past this Illumina read length barrier and really get into questions about composition to function rather than just sequence to function, okay? And so that's this project, which is developing a platform for building and testing synthetic gene circuits, in massively high throughput. So platform I'm going to tell you about allows us to build thousands to millions of gene circuits in mammalian cells, measure them in individual experiments. And the data sets generated by these experiments can be used to train ML models that can actually infer gene circuit composition to function design rules. So I think this is a potentially really powerful approach for solving some of the challenges for part context that have been plaguing the field for so long. And again, just to reemphasize the point, these don't need to, the models that you create from these don't necessarily need to be at odds with biophysical models. They can potentially work together with biophysical models. So the individuals that did this was a tag team effort by Ronan O'Connell and Kshitij Rai, and they have respectively done the cloning and cell engineering and the machine learning and data analysis kind of together as a tandem. And this is a really cool collaboration that I think they really realized in the lab when they were kind of mid-stage graduate students. And I had very little to do with this other than just sort of, we want to make big libraries, guys. They really came to me with kind of the kernel for this project. Both tremendously talented. So this one is up on bioRxiv too. This one's been on bioRxiv for a while. We're undergoing a very, very painful revision process right now. And I think we're seeing the light at the end of the tunnel, but hopefully it'll be in print sometime sometime in 2024. So just to go over this real quick. So the platform's called Classic. And so Classic combines long and short range sequencing to interrogate genetic complexity. And this is something that Kshitij came up with, the acronym that is. So it works as follows. So we, without going into too many of the molecular details here, what we do is we take genetic parts pools and we mix them together with barcode pools, and we do pooled assemblies such that circuit parts associate with each other, and then also each with barcodes. So then what we do is we index the barcode to the circuit using long read sequencing. And so this was kind of one of the new things. And people have done this in the past using PacBio sequencing. Some of you guys may be familiar with that technology. We used Oxford Nanopore to do this. And that required us making some innovations on the barcoding side, but then also on the software side to analyze the sequences. So there's really the enabling thing about this project is right here. So then we also take the circuit library, we put this into cells, and we do some type of phenotypic sorting. And the phenotypic sorting will separate the circuits based on function. And then we do short read Illumina-based amplicon sequencing because you can get so much data out of Illumina sequencing on each of those bins. So this creates a phenotype to barcode index, okay? And then we'll take that construct to barcode index and combine those two and create a map that gives us the identity of the construct, the identity of the circuit, and the behavior, okay? And so the way we make this work in mammalian cells is we have a hierarchical cloning scheme where we take pools of 5' fragments, fragments of a coding region, and then 3' fragments, put those together into promoter, ORF, and terminator pools, and then use those pools to assemble expression unit pools, okay? So the design of the circuit is implicit in how you mix the pools together. And so then what we'll do then is we'll assemble the expression units in the circuit with a barcode pool that's on its own expression unit that makes the transcript that we use to read out the barcode. Okay. And then we'll index those by long read, and then we'll integrate those at single copy into HEC293T cells, run flow-seq, and then do short read sequencing. So that's the workflow for doing the experiments that I'm going to show you here. Okay. And so the test case for this was, what we did for the test case here was we made a very simple, deceptively simple, I should say, SYN-TF circuit. And the SYN-TF circuit here consists of two genes, one that makes the SYN-TF, and the SYN-TF here has an ERT2 domain in it, which when you add 4-OHT, which is an FDA-approved drug, it's 4-hydroxytamoxifen, this will translocate into the nucleus and turn on a GFP reporter. Okay, so in yeast, I would make this circuit. I would think no problem. I can make a really great, extremely high fold change circuit by putting things together with a relatively intuitive combination of parts. But kind of as we and others have found in the field, making a high fold change circuit using this design that's integrated into the chromosome in mammalian cells is actually quite challenging. Say probably, we spent at least 2 years just building these circuits individually, putting them into various types of cells and seeing, 2-fold change, 3-fold change, high background, putting them into different loci. And so, the more kind of we change stuff, it's changed parts within the circuit design, kind of the further we got away from, I think, really understanding what was going on. So there's fundamental rules for circuit engineering in mammalian cells that we totally missed when we were working in yeast before. So that was really the motivation for doing this library approach on this circuit. And so, we just asked if we could use classic to quantitatively profile circuit design space and, really try to find combinations of genetic parts, mostly that we already had in the lab, to maximize fold change activation. Okay. So the collection of parts we used was, was basically stuff, again, that was off the shelf, stuff we were working with in the lab. And we combinatorially varied 10 different categories of parts, ranging from things in the SYN-TF, different protein domains in the SYN-TF, to to composition of the reporter, and then also the coding. And then we also changed the orientation. And it turns out the orientation actually matters a lot, which I wouldn't have thought. And then we put this into our hierarchical cloning scheme. We put protein domains together, and then expression units together, so on and so forth. And we ended up with an indexed library of about 166,000 circuits, okay? And then we put these into cells. We put these into HEC cells, and we did our sort-seq experiment. And here you can see the uninduced library population here. And then here's the induced population. See, some of the circuits are already on, and then some of the circuits never turn on, okay? So there's a lot of heterogeneity in behavior within the library. And then we also got clonal isolates out of the library. And so those kind of acted as our ground truth for seeing whether the measurements that we were making were accurate or not. And you can see here the individually measured clonal isolates, and then their reconstructions based on the sort-seq experiment match up pretty well. And I think this was one of the real surprises in this for me, was just how accurate the sort-seq measurements can be. If you use the right approaches for reconstructing the barcode distributions. And so Classic makes, at least this part of Classic, the workflow for doing sort-seq in HEC cells generates really highly accurate activity measurements. And so I think that's one of the first lessons here, is if you're going to use this library approach, you really need to make sure your measurements are accurate to, within some reasonable error, if you're going to start to think about training accurate models. Okay, so like a lot of high throughput experiments, we weren't able to capture our full library diversity with the sort-seq, and that's perhaps to be expected. We got about 74%, 121,000 circuits. And you can kind of see the distribution of circuit behavior here. So this is the induced expression for the different variants versus the basal expression. See, like most of the circuit library is actually off. It's non-functional, which is kind of consistent with what we ran into when we were building the circuits. There's some that are always on, and then there's some here in this region that are high fold change. So those are the ones we are interested in. And it's not a very big population relative to the entire space. But we were worried we're missing some of the circuits that could be interesting and important. So what we did was we took this data, we used it to train a really simple neural network. And the model showed, so this is the test from a 2080 train test split. Relatively good predictive power with the model. And then what we did with the model was we used it to estimate the out of sample things, circuit configurations that we didn't actually measure. And then we used it to kind of re-estimate a lot of the circuits that we measured in sample. And this is the model, the full configuration space kind of as defined by the model. And what you see is there's a lot of error kind of around the outside between the model and our original measurements, which is to say, like, some of the measurements on the periphery of the behavior space were probably not that accurate to start with. So it's kind of an interesting thing that emerges from this is you can use an ML-based approach to kind of see where data measurements in your data might actually not be super accurate. And then we see actually an even smaller area of design space that's now in the high fold change region. So still circuits, and what we see is interesting. We kind of see now two populations here in this in the high fold change that emerge. And so what we did to make sure that this model actually was accurate is we isolated 100 different clones, individual clones randomly, and we tested them all for fold change behavior. And we found that the fold change for the model was actually in pretty good agreement with the isolated cell lines. And what this told us was kind of the limitations for our design space were actually around 70 to 90 fold, which is pretty good. That's not something that I think we could ever build a priori. Certainly not as good as some of the multi-log fold changes that you see in synthetic circuits and bacterial cells. But it's a good first stab. Okay. So what this gave us now was something that was incredibly interesting for me as somebody who's been working on circuits for a while, and that is a pretty comprehensive profile of circuit design space. So now we can do things like go in and look at what are the part combinations and the part preferences for different regions of the design space. And we actually see there's quite a bit of polarization between things that work in one area of design space and things that work in another area of design space. We took the high fold change region and we clustered it. And we found that there were a suite of a couple different families of solutions that use different distinct part combinations to arrive at a high fold change. And kind of the rules that emerged from that were, I say part intuitive and part non-intuitive. There were some kind of universal rules for what made a good locus design for high fold change. And things like extended spacing between the two expression units, which was probably not that surprising. The orientation of the two expression units was important. And then in the SynTF design, there seemed to be two kind of distinct classes of solutions here. One that favored a really strong activation domain and lower affinity binding of the SynTF, and one that had a little bit higher affinity, but kind of medium strength activation domain. So, you can arrive at quantitatively similar outcomes in this design space with distinct molecular solutions. So that was the lesson here. Okay. So just to summarize this, the second part of the talk here. So, Classic enables an accurate mapping of an entire genetic design space and really allows you to rapidly hone in on circuit compositions that match your target behavior. It's possible to gain insight into complex nonlinear composition of function rules using this approach, which is, again, something that was very difficult for us just building these with kind of our a priori design intuitions. And then these, we're just kind of at the front end of this and, kind of learning as we go here. But it's pretty clear that these large data sets are going to enable really powerful statistical inference and ML analysis that we hope in the future can drive forward synthetic circuit design and really complement what we already have in terms of our biophysical understanding of circuit function. Okay, so the future now, in my lab, this is what the design build test cycle looks like for most of our projects. Not all the projects, but many of the projects are now going to, we're going to try to incorporate some ML model in our process. Building, most of the lab is now focused on building libraries. And I think building libraries using pooled assembly at much higher throughput is, something that makes these projects that are, naturally very slow in mammalian cells go much faster. And then, measuring output by NGS is one of the advantages here. Just all of the data that you get out of a run. It's expensive, but it's also worth it. It's really enabling when you can take this really kind of kludgy approach of moving through design space and just fill out the entire design space in a single stroke. And so we're really working hard to generalize Classic, envisioning workflows where Classic is kind of the central feature of the design build test cycle iteration. And we want to actually, so the project I showed you was about mapping design space. We want to actually try to generate models that can be used to predict design space and forward predict the use of parts that may not be included in the original test space. And then we've actually tried to get Classic to work in a lot of different contexts now, and really trying to get libraries of circuits into a number of different primary cells using an array of different types of vectors. And so the goal with these projects is really going from circuit composition to function, and then to cellular phenotype mapping, and something that we think is going to be tremendously useful for engineering new cell-based therapies. Okay, so I will end there. This is most of the lab. I think there's one portrait that's missing here, but this is the group here. Special call out to Ronan Shittage and Zhao Yu, who did the lion's share of the work that I presented. My collaborators, folks that I have had extended conversations with about modeling and machine learning over the last couple of years. We really kind of entered this as just people that want to do this, and now I think through the intuition that these folks have provided, we're kind of moving things forward now. And then my longtime collaborator Pankaj Mehta, who has a theoretical physics group at BU, who's helped me with modeling, going back many years. And my funding sources, and I'll end there.
Okay, I think we'll have time for a few questions. I'll just start. I have super impressive stuff, Caleb. Enjoyed both arcs. In the phosphorylation story, many of the measurements were made at steady state, but the dynamics, you used this condensate reporter to measure that. Yeah. I was curious whether you were measuring the phosphorylation kinetics, which you said sort of fell in the mid set of timescales, or if you're measuring something about the condensation formation and disassembly kinetics. And then related to that, can the speeds of turn on and turn off be tuned by manipulating the push-pull? Like, can you make it turn on faster by having it kind of more dialed up?
So we can change the association of the substrate with the condensate by changing the push-pull. And I didn't show that, but that was an important control to include, obviously, because you don't want it, as you said, to be the reporter for the condensate and not the circuit. Okay, so I think through that, and there's a couple other lines of evidence, we're relatively confident that is, at least the rate at which it associates with the condensate is reflective of the rate with which it's phosphorylated. Now, if there's an additional rate of, once it's phosphorylated, it needs to go to the condensate and associate with the condensate. That's it's probably true. We have no way to model that at the moment because the measurements we've made, just the measurements you see, right? But I think it's, it's clear that it's within this, temporal range where, it's clearly turning on in the first 5 to 10 minutes. So,
Yeah, I thought maybe it just
checkbox.
might be even faster.
It might be even faster. That's the thing. And if we had a more direct measurement, maybe we could see that. But this is, this is the one we hit on because it was something that we, basically had in hand and we were already working with.
Hi, Caleb. Great talk. I particularly loved the Triple H plots.
Oh,
for its
good. Thank you.
namesake. I had a question regarding them.
Real
So-
WWE fan.
No, the polar vest plots might as well. So one of the questions I had was that it seems like you had the reader-writer, the kinase phosphatase amounts as the two axes. And it seems like you introduced these proteins via transient transfection.
Yeah, good.
But in those Triple H plots, only a certain region of those plots showed functional activity for your system. So I was curious, was that because the regions outside of those plots just don't fall into the right regime for activity, or were there cells there, or were there not cells there? I just had a, and then is it possible to move the functional space from one location to the other, like how Scott said, by actually tweaking the reader-writer themselves?
Yeah, so the parts of the Triple H plot that didn't have color associated with them were mostly because the proteins are being expressed too low in those regions. Okay, so in terms of engineering, you don't want to engineer a population of cells where the circuit's on in some cells and some off. And I hope the reviewers give us a pass on that because it's like a first, kind of a first-in-class demonstration. The real way to manipulate kind of that window of expression is to integrate the circuits at single copy and then control expression levels. So in other words, get away from transient transfection. And we're working on that. That's something we're doing. And we actually have a kind of our first classic library with the phosphorylation circuits that we just generated like in the last month or so. And those are all single copy integrated. And you see those Triple H plots really tighten up when it's a single copy. The value of doing transient transfection is you get a full titration of the components. So there's a lot of stuff I didn't show in the modeling where we're fitting all of the cells in that distribution. So it's just as valuable to see cells that are on because they have enough components and they have phosphorylation as it is off because they don't have, because that allows us to better fit the parameters for the. So that was the fig leaf argument that I offered in the paper, was that we're getting model fit out of this transient transfection, which is a nice trick, as long as we can measure the expression level of the substrate and the kinase, right? Hope that answers your question. Yeah. Yeah. That's now we're getting in the weeds. So this is a problem in flow cytometry, right? Is you have different floors. You can't make stoichiometric comparisons between those, right? So what we did, and I should probably have a slide for this audience about this, is we took GFP and we made a really long tail on GFP. And then we put our epitopes at the end of that tail. And we wanted it to be long so that there was no fret, right? But we wanted it to be, every GFP fluorescence we should be able to see how much fluorescence is associated with our antibodies that are, tagged with whatever dye. And then we related everything in the model, all the concentrations in the model, to each other based on that GFP fluorescence currency. So what the model does is it converts everything into GFP fluorescence, fits it, and then converts it back into antibody fluorescence, and then tells you what the data should look like. So that's the way we navigated that. And it's not perfect, but it's somewhat predictive. It's pretty predictive for behavior. So that's very astute that you caught on to that. That was a big challenge in the modeling. Okay. You trained it well.
Any other questions for Caleb or,
Hey, great talk.
Thank you.
So my question is kind of also about the phosphorylation networks. When you're kind of doing this and mentoring this, how do you compare the phosphorylation networks that might be like accomplishing similar things to maybe networks that have been done before? And by this, I mean the SID-Notch if you're trying to get to the nucleus? How do you weigh the speed and modularity versus like complexity?
Yeah, so Sin Notch is quite slow.
Okay.
Scott knows. Scott worked on Sin Notch. And I think it's it's very much apples and oranges, I'd say. I think the complexity here is much higher, right? I kind of view the 7-gene system as that's kind of the minimum that you need to get into the nucleus and activate. There's probably a couple things we could do to reduce the complexity a little bit from that. But we could make, like, for example, like a single chain receptor, right? And so it's just one gene for receptor. But kind of no matter what, we're looking at like 5 or 6 proteins. So I view that as a challenge. That's the cool thing about Sin Notch. It's very compact. That's why it's in the clinic now, right? This is not going to be in the clinic for a while. The advantage that we have is that all of these, and I didn't go into the detail too much on this, is all of these proteins are derived from native signaling pathways. So all the kinases are just kind of, they're engineered, they're tweaked so that they work the way we want them to. But I don't think any of them are going to produce epitopes that the immune system freaks out over. They're all natural proteins that already exist in human body, that our immune systems are all used to. So I think there's translatability there. The challenge there is the transgenesis challenge that everybody is facing in mammalian synthetic biology. And it's the thing that, when you go to mammalian syn bio meeting, and I go to mammalian syn bio meeting, as I do every summer, half of the talks are tools, and probably a third of those tool talks are different ways to put bigger, more complex pieces of DNA into mammalian cells. So challenge is ongoing. I'm going to say the tool builders are going to catch up with the complexity of my circuits at some point in the next 5 years, and I'm just going to grab those tools and I'm going to put them in, and I think they're going to work. Maybe not as well as Sin Notch to start with, but I think we can design our way to it. Does that answer your question?
Yeah.
Okay.
Caleb, great talk. I really enjoyed it. I was wondering, with your push-pull circuits, are there other ways that you envision expanding those, like to other types of post-translational modification signaling? Or do you think there would be value in, instead of having this constitutive interaction with leucine zippers, making that small molecule regulated so that you have more temporal control over it?
Yeah, absolutely. I mean, I would say, I want to say that we've thought about this, but I mean, I guess really haven't. Anything that we can use to, anything that we can use to program protein-protein interaction that's, say, within the nanomolar range, then I think can be used in the system, absolutely. Other post-translational modifications, again, we thought about ubiquitination, because I think that's, one of the concerns is these, and we see some evidence for this, is the receptors are actually being pulled in into endosomes, and they keep signaling. It's just, something that's, absolutely advantageous for certain types of natural signaling. We don't necessarily want that. We want things to just be reading out the stuff in the environment kind of in real time. So, I think not necessarily installing ubiquitination, but, playing with signals that keep things on the membrane, but then maybe also playing with degradation of the pathway components is something that we're looking at. But I'm open to ideas. I mean, you can put anything on proteins, right?
Let's thank Caleb for a great seminar.
Thanks, Scott. Thank you, guys.
Yeah, I feel like I would love to see the kinase activity in real time. It's actually